Hi, I'm Mr. Williamson, and this video is about diffusion osmosis. Before we get started, make sure you're taking good notes. I have found that Cornell style works for most people. If you have any questions, make sure you ask them in an Edpuzzle or in the classroom. Rewind or to review something, or pause it if you need to write something down. Let's get started. You guys have all experienced diffusion before. You walk in a house and someone's cutting up an onion and you can smell it because of diffusion. Your dad has blamed the dog for something and it diffuses out and smells up the room and the dog is blamed. Diffusion. May have an oil diffuser, essential oil diffuser, which basically puts the oil out in one area and it diffuses and makes the rest of the room smell good. Maybe you use it after blaming the dog. Diffusion. We experience diffusion when we leave school. When the bell rings at the end of the day, we have this is King right here, and everybody starts leaving school and going down the streets and exiting. Actually, they don't exit that way. They all exit out the front and they all go out into the neighborhoods. Okay, they're starting out in one area and diffusing out from the school. We do that all the time. It's diffusion at the end of every day when the bell rings. Diffusion is caused by a gradient. All right, and a gradient is a gradual change from a high concentration to low concentration. So here you're seeing a gradient from black over here to white over here. All right, and as we go through that gradient, it changes gradually. So if we're looking at the concentration of black, we would be looking at real high concentration over here and a very low concentration over in this area, and actually none over here. So there's nothing over in that area. So it's a gradient. Basically, it's a concentration gradient. We're going to go from a high concentration to a low concentration. All right? So we start from a high concentration here, all right, where it's greatest, and then as we go down, back to that white, the black and white, as we go down, we go to a lower concentration. All right? So it's a high to low, moving from a high area of concentration or an area of high concentration to an area of low concentration. That's why when you walk into the house and someone's cutting an onion, you can smell it. The constant area of high concentration is right where the onion is. That's the highest concentration. But then as it moves out, it gets lower and lower, and it's diffusing out. The aroma, the chemicals that cause the onion smell are diffusing out from that high area of concentration, the onion, to a lower area of concentration, wherever you are. Same thing happens when we're talking about school. We have at King, we have a really high area. When we're normally in school, we have 3,000 students between the ages of 14 and 18, all concentrated there until 250 or whenever we get out of school. And at that point, we all diffuse. We go to a low area. All this out here is a low area of concentration. During school hours, the number of teenagers between 14 and 18 is really low in those areas, really high at King, and then it diffuses out. They all leave, but they don't all go in the same direction. They're all going different directions into the different neighborhoods and etc. So they're diffusing away from the high concentration. We can do the same thing with molecules and atoms. They diffuse. They go from a high concentration over here to a low concentration over there. And they will move gradually from the high concentration to the low concentration. That's what's happening with the onion. It's the smell, the onion smell, the aromatics of the onion molecules are diffusing from a high concentration to a low concentration. Why does that happen? Because molecules are moving, constantly moving. And as they move, they can be carried farther and farther away from that high concentration. Okay, that's called diffusion. All right? It's the molecules in a solution spread over time. All right? They will diffuse out. So we could look at it this way. We start with time 1, All right, this is T1. This is, let's say this is right after you cut the onion or something like that. We can go to T2, where they spread out a little bit more, and then we can go to the third time period when they spread out even more. So you can see that the molecules, or the dots in this case, are spreading out, going from an area of high concentration to an area of low concentration. Or we can look at it, same picture, just different way of viewing it. We have them spreading out and continuing to spread out and then continuing to spread out. And they will continue to spread out through diffusion, moving from a high area of high concentration to an area of low concentration. So what is it exactly? Well, diffusion is the movement of molecules from an area of high concentration to an area of low concentration. I've been saying it for like the last 2 minutes. Moving a substance, molecules, from an area of high concentration to an area of low concentration. Now we can affect the rate of that. We can change how fast that happens based on the size of the molecule. Bigger molecules don't move as fast. Smaller molecules move faster. We can talk about the pores in a membrane, which is what we're going to do here in a minute or two. We can talk about the temperature. The higher the temperature, the faster the diffusion rate. Because when you heat things up, the molecules move faster. That's what happens when you add heat to a substance. We can talk about pressure, adding pressure, or like a vacuum. That's how a vacuum cleaner works. It decreases the pressure in the vacuum and it pulls the things in. Not really diffusion, but the same principle. Or the concentration. If you have a very, very high concentration at the one area and a very low concentration in another area, that'll diffuse faster. So the factors that affect diffusion, size, like that we're going to talk about, size of the molecules, the size of the pores, temperature and concentration, not so much pressure in biology. So here's a video demonstrating the effect of temperature on diffusion. All right, you've got warm water over here, you've got hot water here, and you've got cold water here. Now the cold water looks a little blurry because the condensation on the outside of the glass makes it blurry. So here we go. So what we're going to do is add a couple drops of food coloring to each one, and you'll notice how quickly the warm water changes. Okay, how quickly it diffuses, the hot water. The warm water over time is diffusing a little bit quicker over here, and the cold water isn't really diffusing at all. It's just dropping down. Add a couple more drops, and again, you see how quickly the hot water diffuses because the molecules are moving very, very quickly, whereas the cold water, not. Okay? After some time, you can see the change. Warm water is slowly diffusing, cold, not at all. All right? So temperature is a huge effect. All right, so diffusion is going to continue until this thing called equilibrium is reached. It's a word you probably should know, equilibrium. Equilibrium means that an equal distribution of the molecules. So we went from a high concentration over here. Over time, that started to spread out, and you can see that they were looking at the red, until it is equally spread out over the entire thing. All right? When equilibrium is reached, there's no more diffusion because there's still movement. Things are moving. The molecules are moving, but there's no spreading out. They've gone as far as they can. A great example of this would be kind of like a parking lot. If you have space in the parking lot or this parking structure, the cars can keep coming in. So they'll keep going in until you reach a point where the parking lot or the parking structure is full. That doesn't mean cars can't be moving, but there's no place for them to park. Equilibrium is reached when one leaves and the other one parks. And one leaves and another one parks. And one leaves and another one parks. That's equilibrium. Up till then, cars can keep coming in, keep coming in. Don't get me wrong, cars can leave, but cars will keep coming in. You can have it's like diffusion or the movement of the cars into the parking structure until it's full. And then we can only bring a car into this parking structure and park it when one is left. That's equilibrium. One has to leave before the other one comes. All right? So what I'm going to ask you to do here is predict what will happen to the different molecules in the figures that I show you. All right, so you have here a membrane which has little openings in it. You have two sized molecules. You have a larger molecule and you have a smaller molecule. And I'm asking you, what do you think is going to happen? Okay, how is it going to diffuse? So go ahead and answer that, and let's take a look at the answer. All right, because of the size of the holes, the red ones can't get through. They're blocked. But the smaller ones, the blue ones, can. So equilibrium is reached by the blue, not by the red. All right, so you have an equal number on both sides. The blue will pass over until equilibrium is reached, meaning that maybe this blue is going that way, but at the same time, that blue is going the other way. Equilibrium. All right, so another one. What do you think is going to happen here? All right, answer. Again, equilibrium. Unfortunately, there's only 3 blue ones, but this one is, you've balanced it out. What will happen is probably this one will pass back over, this one will go that way. Equilibrium. They're still moving, but they're moving at the rate where they pass both directions at the same rate. Okay? Another one. What do you think is going to happen? You've got 6 red ones, and the holes or the pores in the membrane are a little bit bigger. So the answer, equilibrium. The red is actually able to pass through the membrane until it gets to the point of equilibrium, an equal number on each side. Again, remember, they're still moving. Once they've reached equilibrium, doesn't mean they stop. It just means that one going one way is the same as the one going the other way. Okay, here's another one. What do you think is going to happen? Some much smaller pores. Okay, well, nothing. There is no change because they can't pass through the pores. That's those are the type of things that happen in biology. Is we're looking for equilibrium, balancing out the amount, the concentration of the molecules. Osmosis is a really special kind of diffusion. Up till now, what we've been talking about is balancing out the concentration of the solute. Remember the first unit when I talked about solutions, you have a solvent and a solute. The solvent is the part that is dissolving something. The solute is the thing that's being dissolved. So if you're making salt water, water is the solvent, salt would be the solute. All right? And diffusion doesn't have to have a membrane. Like I said, the onion. You're cutting open the onion and there's no membrane involved. It just diffuses throughout the house and makes your house smell like an onion. All right? Osmosis is the diffusion of water. Okay? I think it can actually be a diffusion of something else, but in our case, it's the diffusion of water. And it's going across a selectively permeable membrane. So it looks something like this. We have a concentration of water, okay, over here that is much higher. And you can see the concentration in this case is represented by the little blue dots. All right? The little concentration over here is lower. So you have high concentration on the left, low concentration on the right. The direction of the movement of the water is going to be through a membrane, is going to be from a high concentration to a low concentration through the membrane. The other particles are not. The other particles outside the cell could be salt. Inside the cell, they could be proteins. Doesn't really matter what it is, but there's a concentration of water compared to the solute, the thing that's being dissolved. This is so powerful that we can actually move water and raise it. And this is, I can do this in class when I have the equipment, but I can't do it here. So this is probably the best demonstration I can give you. If you start out with water at one level, right, an equal level on this side, we have a semi-permeable membrane, all right, that's right there. So we can see that the semi-permeable membrane goes down the middle of this container. And on the left side, you have, and I'm going to make up the numbers, let's say it's 100% water. And on the other side, let's just say it's 70% water. What's the other 30%? Salt molecules. So this is a 30% salt solution. The water is going to go, the water, not the salt, the water in osmosis, the water is going to move from the high concentration, 100%, to the low concentration, 70%. As a result, the water is going to actually level on the left side is going to drop down because it's moving over to the right side, and the water on the right side is actually going to go up. We can actually lift water up the water column because of the pressure of this. All right, so, but it's the water, it's osmosis going from a water going from a high concentration to a low concentration. The difference between osmosis and diffusion, all right, and I'm going to leave this here for a minute or two as I talk about it. In diffusion, we're talking about the particles moving. Water could also be part of it, all right? Like you could have a glass of water or a bucket of water and it evaporates, and that's technically is diffusion. It's going from a high concentration and evaporating, and it will continue to spread out until it fills whatever area that's in, all right? And so we're talking about the solvent and solute of particles moving, and there's no semi-permeable membrane. In osmosis, only the solvent moves. This would be water. So only the water's moving, not the salt, not the sugar solution, the sugar that's in the solution, but only the water, okay? And it does have a semi-permeable membrane. It has to prevent the salt or the sugar from moving or whatever that we're talking about. Both of them, what the goal is to equalize the concentration of the solutions or the concentration of whatever we're talking about in the two areas. So like the onion, ideally that onion would spread out until all the onion smell molecules are equally distributed throughout the house. That'll never happen because the onion's in the kitchen. All right, but that's the goal, is to equalize what the spread out of whatever we're talking about. And in osmosis, the diffusion of water, we're trying to equalize the concentration on both sides of the membrane. Probably the best example for me to give you for osmosis occurs in blood. All right. Now, we talked about the surface area of a red blood cell and how they found that in the cell membrane video. But we're going to go back to it because they're kind of cool. So here's a drawing, and these are actual photographs of a electron microscope photograph of red blood cells. Red blood cells react differently in different types of water. Salt water compared to a high concentration of salt water compared to a pure water, or what we usually have. I think it's 7%, I think, or something like that. Anyways, so here's the three different types of solutions you could have, and I'm going to explain them to you because you need to know this. You need to know this. A hypotonic solution is the inside of the cell is like, let's call it 25% water. All the rest of it's proteins and salts and sugars and things like that. Outside the cell, call it 75% water. All right? Which way is the water going to go? Well, the water in this case is going to go into the cell because it's going to go from a high concentration of 75 to the inside where it's low, 25% concentration. Okay, that's hypotonic, and I'll explain it more in detail. Isotonic is equal. So you've got inside the cell, you've got a 50% solution, and outside the cell, you've got a 50% solution. Which way does the water go? Well, remember, it goes both ways, equilibrium. So it's going in from 50 to 50, but it's also going out at the same rate, 50-50. So there's no change in the cell. And then you have a hypertonic where the inside of the cell is more concentrated water, 75%, and the outside of the cell is a low concentration of water. Maybe it's really, really salty water or something like that. Well, in that case, the water is going to go from inside, from a high concentration, to a low concentration. So the water is going to leave the cell, and that causes the cells to shrivel up. All right? So these are the three blood cells, again, under an electron microscope, showing you the three different results. Let's take a look at them. If we look at an isotonic concentration, it's the same inside and out as it is outside. And I'm making this up, the numbers up, but let's call it's 50% inside and it's 50% outside. Okay? So which way, what's going to happen? Well, the water's going to go, it's going to go out of the cell, and it's going to go into the cell at the same rate. So there is no change. That's called an isotonic solution. Iso means the same. All right, you need to remember that, and I've got a way to help you remember that. What happens if the concentration is higher outside the cell? So let's say that it's a 100% solution, water solution. It's pure water, all right, and it's only back to the original 50% inside the cell. Well, the water would go in. That causes the cell to swell. That's called cytolysis. So a hypotonic solution causes the cell to swell up, all right? And that can cause cytolysis. Cytolysis is when the cell actually bursts. It's like blowing up a balloon so much that the balloon bursts, all right? The way I remember hypotonic is the whole key, all right? There is hypertonic, which I'm going to talk about here in a minute, all right? But hypotonic, I think of this as a giant O. When we're done with this cell, it makes a really, really big O, which is in hypotonic. Hypotonic cells swell up to make a large O. How do you make something swell? You make the water go into it. So the concentration must be higher on the outside than on the inside, so it swells up to make that O. Hypotonic is the one that has the O, makes the cell swell up. The third one is hypertonic, and I have this wrong on the slide. These should be reversed. This is the higher concentration is actually not outside, it's inside the cell, and it's not inside, this should be outside the cell. My bad. But basically we're talking about a concentration, again, of maybe inside the cell it's 50% inside, but it's only 25% outside, all right? So the water is going to go out. It's going to go from a high concentration to a low concentration. All right. The result, the cell shrivels up. That's called plasmolysis. So you've got three that you need to remember. And part of learning is you're learning tricks to remember things. You have three things you need to remember, and they are starting down here. Let's call this hypertonic, isotonic, and hypotonic. How are you going to remember which ones those are? Like I said, the one that has the O is the one that makes the cell into a big O. That means that the water has to be going into the cell. All right, now you're going to have to remember hypertonic, isotonic, and hypotonic. You're going to remember the terms, but which one does which? Hypotonic swells the cell up. Hypertonic does the opposite, shrinks it down. And isotonic keeps it the same. Hopefully, maybe that'll help you remember. If we put this into effect in biology, we got two plant cells. These are Lodia cells. They're a plant that you can get at your, PetSmart. You put it in your aquarium. This on this side over here, I'm not as concerned with this one because it looks very similar to a regular cell. But when we put one of these, a leaf of this into a really, really strong salt solution, all the water leaves the cells. Well, that causes the cell membrane to pull away from the cell wall. Remember, the cell membrane is inside the cell wall. So what happens is you have it shrivel up, like over here. It shrivels up, pulls away. Now, if we put it back into regular water, fresh water, it will swell back up because it'll be hypotonic and it'll cause the cell to swell. But if we put it into a really strong sugar or salt solution, it shrinks up, hypertonic. So it affects plants. And we can also, you can do this at home. You can do, this is what I did one time as an experiment to demonstrate it. If you take gummy bears, and they have to be, I think, the original gummy bears, which I forget the name, Harbo, maybe, something like that. And you put it into a salt solution, vinegar, water, iced tea, soda, or rice milk, you can see the different shapes. With a salt water shrank. Why? Because the water actually leaves the gummy bear. In water, it swells up. And you can see, but why does it do that? Because of osmosis, the water going from the high concentration to the low concentration. So, let's take a real quick review. All right. We talked about diffusion, the movement of a substance from a high concentration to a low concentration. We talked about the concentration gradient, which is a gradual change from a high concentration to low concentration. We talked about why this occurs, because atoms and molecules are constantly moving. The more higher the temperature, the more they move. We can also change it with the size of the pores in a membrane. We talked about what diffusion was, the movement of something from a high concentration to a low concentration. And we talked about equilibrium, where they're equally spread out throughout whatever area that we're measuring. All right, and that could be for both osmosis or diffusion. We talked about osmosis, which is the diffusion of water across a membrane. That's important. It's across a membrane. And we talked about three different types of solutions. A hypertonic, where the concentration is greater inside the cell than outside, so the water would leave. That causes plasmolysis, okay, the shrinking of a cell. Isotonic, where the concentration is equal, so the cell doesn't change size or shape because the water is passing back and forth. And we talked about hypotonic. Hypotonic is where the concentration is greater outside the cell than inside. The water goes into the cell, swelling up. Hypotonic, and it can cause cytolysis. Cytolysis is where the cell actually bursts. So again, if you have any questions, make sure you ask them on the end of the Ed puzzle or ask me in class. Hopefully you took good notes. Have a good day. Good luck.